Bring everyone in.
Welcome, everyone, to our second session, I guess, on ecology and evolutionary ecology. I'm Howard Rundle, University of Ottawa. I'll be moderating this session, and we'll get right going to stay on time. A reminder to the speakers, they've got 15 minutes total. I'll give a checkmark warning at 13 minutes and flick on my video at 14. And if you're not wrapped up by 14 and a half, I'm going to pop in to cut you off so that we have time to set up the next speaker and stay to time. So I'll turn it over with that to our first speaker, Antoine.
Thank you. Oh, we should start at the beginning. Yeah. Yes, I'm Antoine Mellet. I'm a PhD student in Freiburg, Germany, and I will present an experiment in which I try to select against sociality in a cooperative breeder. And I will start with a presentation of the model species because it's not your typical social insect. Xyloborhinus saxicini is a coleoptera, subfamily Scoliginae, and they live deep in dead wood. They dig tunnels deep in the xylem, and they do not forage for food, so they are difficult to spot in the wild. But we can breed them in artificial medium made mostly from sawdust that we pack in tubes, and they dig these chambers with a few tunnels, and that allow for observations and experimentations in the lab. So the life cycle of the species starts when a single mated female lands on a suitable piece of wood, and she digs the tunnel in the xylem. She lays eggs, and they develop as larvae, pupae, and then emerge as adults. And they do not leave their native nest immediately. They stay for several days, sometimes weeks. And they mate, brothers and sisters, in their native nest. They do not lay eggs in their native nest. Only the founding female does that. And the foundation, the dispersal and the foundation of a new nest is the most critical step in the life cycle. There is a high failure rate. But once the larvae hatch, the nest is almost guaranteed to produce adults of the next generation. So the combination of a highly risky dispersal and the high relatedness within the nests, that set the stage for cooperation and altruism that we indeed observe. The beetles cooperate to dig the nest, to keep it clean. They also groom each other. And that, combined with the reproductive skew and the overlapping of generations, that qualify Xyloborhinus saxicini as a cooperatively breeding species. That's a relatively old nest. You see that there are several adults and they are still present. They can reproduce, and they will all eventually try to found their own nest. They will disperse from their native nest and start their own, but they don't do that on the very same day. Some disperse early and some disperse late. And we have evidence that this difference in timing of dispersal correlates with different reproductive strategies. That the early dispersers, they have a more selfish strategy, and they focus on their direct fitness. And the beetles dispersing late, they invest more in the group, and they gain a larger part of their fitness through inclusive fitness. So I made the assumption that this difference in the time of dispersal, this difference in reproductive strategy is heritable. And by controlling the conditions in a lab experiment, I could isolate the genetic component of this variability. And I had two hypotheses that by selecting early dispersing females over successive generations, then I will have adult females dispersing earlier and earlier. And second, that would correlate with a decrease in the cooperative behaviors. To test that, I split the lab-bred population in two lineages, a control and a treatment. In the control lineage, I selected dispersing females at random and used those to start the next generation. And in the treatment, the early disperser treatment, I only picked the first females to disperse from any given nest, and only those females I used to start the next generation. And I maintained this selection regime over 5 successive generations. From these different groups, I recorded the time of dispersal, so how many days between the very foundation of the nest till the dispersal of the offspring. I recorded the success rate, and I recorded the number of offspring dispersing from each nest, and I also recorded the behaviors, especially the cooperative behaviors, and both of the larvae and adults, because the larvae are active, which is quite rare in social holometabolous insects. I will not present the whole data set. I will focus on the first and the last generation from each lineage. I had a number of successful nests, and the first interesting thing is that at the 5th generation, the dispersal happened earlier than in the first one. You see that the dotted line, they are from the first generation, and the solid lines, they are from the 5th generation. And as you can see, the dispersal happened earlier in the 5th generation. The difference was not significant for the treatment lineage, but it was significant for the control lineage. So my selection regime did not had the expected results, but I still noticed some patterns and some trends that I think are interesting, and I will present that. The first thing is that the success rate increased in both lineages. It more than doubled, and yes, was around 25% during the first generation, and was over 50% for both lineage at the 5th generation. The number of dispersing females per nest was different in the first generation, not significantly different, but there is a trend for converging of this productivity. And also the behaviors varied. So the larvae never cooperate that much. They don't invest that much time and effort in cooperation, but still it was higher in the first generation than in the 5th. But for the adults, I found no such pattern. The cooperation was always low and also very consistent and very stable. No difference between the two lineages, and no difference between the first and the fifth generation. So to summarize my results, I made the hypothesis that adult females will disperse earlier and earlier. They do. The difference was not significant in the treatment lineage. And I made the second hypothesis that the cooperative behaviors will decrease in frequency. They do, but the difference was not significant either. Combined, these two patterns may suggest that the pressure for cooperation is relaxed in the laboratory condition, and that may explain why the cooperative behaviors are less frequent at the end of the lines. Also, interestingly, the success rate increased, so we are sort of breeding super beetles in this experiment, and also the groups that dispersed the later, they also had the highest productivity and they had the most offspring. That correlates nicely with other pieces of evidence we have on this species, and probably the feedback between the time of dispersal and the social environment and the productivity of the nest, probably this feedback and this correlation is at the core of the evolution of sociality in this species. So with that, I would like to thank my colleagues from the chair of Forest Entomology and Forest Protection. And of course, thank you for your attention. And if you have questions, I will be happy to answer.
We have time for questions. Feel free to raise your hand or add it to the chat. I don't currently see any hands, but I had one question myself. So the increase in survivorship that you observed, could that be adaptation to, either the lab or a novel maintenance protocol that changed? Like if the normal maintenance routines changed during experimental evolution, then could populations simply have been adapting to either general lab conditions or that changed maintenance routine, and thus they're just increasing performance across all groups?
So the control lineage in my experiment, the condition, I try to stick as close as possible to the lab breeding routine. That may be adaptation. That's also, I think, side effect of just breeding. Of course, only breeding beetles participate to the next generation, so that over time, just that selects for high quality beetles. So that's definitely a bias, but I think that's the risk with any lab bred population.
Well, presumably, if the population from which you derived these had been maintained in that same way, most of the response to that would have played out already. Interesting.
That's a trend that we observe. We capture wild beetles every now and then to refresh the lab population, and that's a very common trend that the success rate increases over generations.
Any other questions from the audience? If not, we'll get our second speaker set up so they can start on time. Thank you for an interesting seminar. Yep, we can see your slides. We'll just give it a second here till the clock ticks over in case anyone's coming in from moving between sessions. Keep us on schedule.
Okay, sounds good. Can you hear me okay?
Yep.
Great.
Okay, take it away. Ian Osprey is up next.
Hello, everyone. My name is Ian Osprey, and I'm a postdoctoral researcher at the University of Bern in Switzerland. And I'm really excited to talk to you all about some work I've been doing for the last 4 years, looking at how eye size contributes to the ecology and evolution of the avian tree of life. And as I'm sure most of us know, birds live across an enormous gradient of light, right? So birds inhabit all kinds of habitats across the world, ranging from very dark, enclosed environments such as the interior tropical forests or coniferous forests in the temperate north, to fairly open terrestrial environments like deserts and grasslands, to then aquatic environments that tend to be very open and brightly lit. And birds have obviously adapted to live across these gradients of light by adapting a large gradient in eye size variation. And anybody who spent any time looking at birds in the field knows that birds vary a lot in their eye size. And the reason this matters is because eye size strongly predicts both visual acuity and sensitivity. So visual acuity is the ability to resolve details of an image, while visual sensitivity is the ability to distinguish an object from its background. And Eleanor Cave just came out with this really great paper this spring showing that when you look at the relationship between eye size is measured by the axial diameter, and axial diameter is basically the length of the eye from the front to the back. When you look at that correlation and you correlate it with actual behavioral and cellular metrics for acuity, there's this very strong correlation between eye size and visual acuity. So eye size can be considered this very strong proxy for basically the ability for a bird to resolve details of an image in its environment. And so, for example, she's created this great R package that lets you kind of simulate how visual acuity really ranges across different species of birds based upon their eye morphology. And you can see this really matters. These two different species of birds, Anna's hummingbird and Eurasian magpie, really vary a lot in their acuity, and they're able to resolve their ability to resolve an image in the environment. But the important thing to understand is there's also exists this metabolic performance trade-off between eye size and then metabolic investment. Eyes are very metabolically expensive organs to maintain. So as eye size increases, it requires an increased metabolic investment. And so, this means that we would expect there to be the evolution of these kind of adaptive optima given different morphological and physiological and ecological constraints. And so I've been really interested in trying to understand how does eye size variation really contribute to the ecology and evolution of bird communities across the avian tree of life. And I got started actually on this question in kind of a random way. I was doing my PhD work at the University of Florida, and I was very much interested in identifying different functional traits explain why different tropical bird species disappear in degraded landscapes. And I was working in the Andes of northern Peru, and this is a flame-faced tanager that's an iconic species of the Andean mountains that's adapted to very large amounts of light forest canopy. And I was very interested to understand basically how birds adapted to different light microenvironments across the light spectral gradients that you find within the cloud forest in the Andes. And while this tanager is adapted to very large amounts of light, there are these understory species, such as this species of ant pitta that basically only lives in very dark environments at the canopy floor of the forests. And I was really trying to test this light sensitivity hypothesis, trying to see how tropical birds show interspecific variation in the use of different light microenvironments, and then can these adaptations actually predict sensitivity to disturbance? I did this work by attaching these light sensors to the back of a bunch of wild birds, over 70 wild individuals, and I tracked them in the field, and the light sensors fell off the birds, and I was able to actually download the light data that they were collecting to see the exact light intensity environments that the birds were using. And I came up with this really beautiful graph that shows basically that as you progress through the day, birds are stratifying their use of different light intensity microenvironments depending upon the species. And then some species are using these really bright environments, whereas other species are using dark environments. And you'll also see there's a strong correlation between the light intensity and where they're foraging, whether the ground or the canopy. So what I did after this is I was also measuring eye size across a wide diversity of species across this forest bird community, and I extracted the residuals of eye size versus the relationship to eye size and body mass. Is a strong allometric relationship between the two metrics. And basically large residuals, positive residuals, indicate kind of larger eyes than you'd expected given their body size, and negative are the opposite. They're smaller eye sizes than you expect given their body mass. And we found this really, really beautiful pattern where species that use these really bright light environments you see on the y-axis tend to have smaller eyes, whereas species that really use these darker environments, they tend to have larger eyes. And this is really the first study to show this functional link between eye morphology and the type of light the birds are using in their environment. And so what I did at this point is I scaled it up to the full forest bird community by looking across this kind of land use light intensity gradient ranging from very dark interiors of forests to very bright silver pasture environments. And I did a whole bunch of point count surveys. These are audiovisual surveys quantifying the entire bird community across this agricultural land use gradient. And I found this really nice pattern where basically a lot of the species that are declining in abundance or disappearing from these very disturbed landscapes tend to have larger eyes. And you see this pattern where species with larger eyes tend to be declining in abundance compared to their abundance in forests. And we really hypothesized this had a lot to do basically with their adaptations to light microenvironments. So these species living in these very dark environments are basically maladapted to really bright disturbed landscapes. So when I was doing this work, which was published in Ecology a couple of years ago, I came across this very interesting opportunity to expand this work to a larger scale and to really ask this question, do these patterns hold across the avian tree of life? And so I came across this unpublished dissertation that was written by Stanley Ritland 40 years ago in 1982. And he basically measured eye size from museum specimens for nearly 4,000 species of birds, or roughly a third of the avian tree of life. He traveled across museums, taking these measurements from specimens produced of alcohol, and then basically left science and never published it. And I was able to pay a couple of undergraduate students to help me digitize these hundreds and hundreds of pages of data that he had collected and make them usable. And I'm still in the process of working this data, but this is the first publication that came out of this. I did a large comparative analysis looking at nearly 3,000 species of terrestrial birds. Again, this is about a third of the terrestrial avian tree of life, looking at ecological correlates of eye size. And basically what I found out is that birds with large eyes or increased visual acuity, they tend to use long distance foraging maneuvers. They tend to eat invertebrates. They tend to live in forests, and especially in the tropics. The tropics tend to have species with relatively larger eyes given their body mass. And the other really cool thing that came out of this is that I basically compared all of the eye traits against a whole bunch of other morphological traits that are traditionally used in these kind of ecomorphological analyses, ranging from wing length to tail length to tarsus length and different bill dimensions, and basically standardized all of the measurements so they're on the same scale, and then put them into a big regression and found that, there's a bunch of different ecological traits that are very highly correlated with eye size compared to all these other traits. Things like habitat. Eye was the top-ranked explanatory trait for whether a bird lived in forest or not. Third-ranked trait for whether it used these kind of long-distance foraging maneuvers or not. Again, related to latitude, that was another big one, and then a diet and dietary traits. And in general, eye size adds as much as 9 to 29% more variation explained on top of all these other traits. So eye size is really this very powerful trait that adds to our information about ecomorphological relationships, as well as predicting sensitivity to disturbance. When we look at this from a more evolutionary perspective, I found that about over 60% of variation eye size alone is explained by ancestral relationships. So, there's a lot of signal. You can see that in the tree, right? There's a lot of kind of clustering among different families in terms of residual eye size variation after controlling for body mass. And you can see here, I did both phylogenetic and non-phylogenetic analyses. And when you invoke evolution, it just explains the vast majority of variation in eye size across the terrestrial bird tree. So there's a lot of interesting patterns here that could be worked in the future on the exact evolutionary dynamics going on in relation to eye size variation. This work from a new paper that's going to be coming out soon in Journal of Animal Ecology. I extended this work to look at aquatic bird species. Now, you think of aquatic birds, they live in extremely open environments where they're exposed to just constant large amounts of bright light, And what's really interesting is, you would expect these species would likely invest a lot less metabolic energy and adapt in evolving larger eyes simply because they don't have to. It's very bright in conditions. And so having large eyes perhaps is not necessary given the light that they're living in. And this is actually what I found when you look at the allometric relationship between axial length or eye size and then the mass of the bird species. All the aquatic species in blue, you can see have this shallower allometric relationship compared to terrestrial species. And then if you look at residual eye size, so eye size relative to body mass, you find also terrestrial species tend to, on average, have larger eyes given their body mass than aquatic species. Again, supporting this idea that this group of birds that live in these very bright environments basically don't need to invest quite as much of this metabolically costly organ. Likewise, I found similar patterns looking across broad foraging types. These are stalkers. These are like herons or egrets that identify prey and then strike them. Species that dive in marine environments, they have to visualize prey from as much as 20 to 30 meters above the ocean. These groups of birds have some of the largest eyes, likely because it increases visual acuity. Whereas underwater pursuers, these are like auks and penguins and things, they tend to have smaller eyes. Again, species using these longer distance foraging maneuvers, hyperopic maneuvers, they tend to have larger eyes. And of course, nocturnal species also tend to have larger eyes. And this was really cool too. I just looked at kind of coarse metrics for plunge dive depths. These are species like gannets and boobies and frigate birds and tropic birds that basically hover 10, 20, 30 meters above the ocean and then plunge. And the larger eyes the species has, the higher it tends to dive, again, suggesting the higher dive heights are driving, increases in visual acuity. And likewise, I looked at the Penguinus database of dive depths and these loggers that measure how deep oceanic species dive underwater. And as you get dark, as species dive deeper into darker oceanic conditions where there's less than 10% of light, they tend to have larger eyes, likely because it increases visual sensitivity as well. So these were, I thought, just really cool kind of intuitive results to find. Similar to terrestrial birds, around 60% eye size variation is explained. Hagel's lambda is around 0.94, so a lot of signal, a lot of clustering going on across the avian phylogeny. And then finally, I had this nice opportunity to work with Mark Hauber and this nice collaboration. We looked at the reproductive consequences of visual systems, in particular brood parasitism, which is this phenomenon where basically a brood parasite is species that doesn't make its own nest. Instead, it lays its eggs in the host species' eggs so that the host raises its young. Instead, we found that hosts tend to have smaller eyes on average compared to their parasites. And this is especially the case for species that have non-mimetic eggs. These are eggs that look different from their host species. So these brood parasites that have very different looking eggs, they can have much higher rates of egg rejection. The host is much more likely to reject their eggs. And this is related to eye size as well, suggesting that even the visual system of birds is very much tied to the reproductive ecology as well. But just to kind of conclude up quickly, I found these patterns or avian eye size strongly predicts microhabitat use and sensitivity to disturbance, foraging behavior. If you have a larger eye, you tend to use these more long distance foraging maneuvers that are better able to identify distance prey, more likely to eat invertebrates. Again, harder to identify an invertebrate in the environment compared to a fruit or a seed. So improved visual acuity is going to help with that. Macroecological relationships in the tropics, there are larger eyes, increased susceptibility to brood parasitism because hosts tend to have smaller eyes. And just in general, eye size is this powerful functional trait underlying the ecology and evolution of birds. And I would love to work with a collaborator who knows how to do a lot of these kind of evolutionary dynamic analyses to really understand more in the details of how eye size has contributed to things like diversification rates or other evolutionary dynamics. And I'd be really interested to talk to somebody who would like to work on that project in the future. So thank you all for your time and attention. And I'm happy to take a quick question before the next speaker.
We may have time for one quick one.
There is a question in the Q&A.
Yeah. Oh,
and we also have a hand raised.
How is eye sensitivity involved in these relationships? Not quite sure what relationship.
Yeah, I think I talk a lot about visual acuity and less about sensitivity in part because I'm just aware of a lot less research on the topic, especially across large numbers of species, right? Because that's getting into like the cellular structure, the anatomical structure of eyes, and there's a lot less data on that, but that's something I think it'd be very interesting. I think in particular, that's why I found this relationship between forests, where forest specialists tend to have larger eyes. You can imagine the darker forest environment, that increased sensitivity could be important. But I think teasing apart the acuity versus sensitivity is definitely something that needs to be worked on more.
I'm not sure we have time for another one. We're 1 minute. Sorry, Joe, you can always put it in the chat and we can continue that.
In addition to the chat, there's also a Q&A on Zoom, and there's a couple questions in there too, Ian. If you'd like, you can just type the answers directly in there.
Okay, cool, great.
And anyone who didn't get to ask a question can ask it in there.
Great. I've been told our next speaker is a pre-recorded talk. I'm not sure if they are in the audience right now to answer questions on the chat. If you are, maybe just speak up in the chat so we the audience knows that they can ask questions there. Otherwise, we'll queue it up and get it started in a few seconds.
All right, yeah, I don't see Blanca in the chat, so I don't think she'll be
I
available.
didn't either, unless there were sometimes the Zoom name might differ from.
All right, I'll start playing the video. If there's any issues, just let me know.
Good morning. I'm Blanca Chavez Sandoval, and I'm presenting the work of obtaining chromosomal introgression in a homozygous state reveals the genetic architecture of reproductive isolation barriers. In this slide, I present the polychain chromosome squash technique used by to identify the introgressive segments of a chromosome from one species to the genetic background of another species. In panel A, we have two homozygous chromosomes from a cross between pure species. In panel B, polychain chromosomes with asynaptic structures are observed that break the pairing between homologous chromosomes of F1, descendant from sister species Drosophila kefere and Drosophila busati. In panel C, a single introgressive segment is observed in an asynapsis of an on-spring in F15 backcrosses. This is the method of obtaining introgressions in the heterozygosis. As Darwin pointed out, in interspecific reciprocal crosses often differ in whether or no fertilization occurs in the viability of fecundity of F1. This pattern of asymmetrical intrinsic polyzygotic isolation was being called Darwin's corollary to Haldane's rule. Darwin's corollary requires Haldane's epistatic interaction. In case 2, reverse crosses between the Kefere females and the Busati males produce fertile females and sterile males. In case 3, hybrid crosses between hybrid males with hybrid females do not produce descendants. Either in case 4, but crosses between the Busati males and hybrid females also do not produce offspring. In case 5, reverse backcross between parental D. Kefere males with hybrid females produce fertile females and sterile males, similar to case 2. In this study, we obtained for the first time homozygous introgressive segments for the pair of sister species, the Busati and D. Kefere. Panel A describes the strategy of crosses and backcrosses to obtain introgressive segments in heterozygosity. In panel B, homozygosis. This is the results of sterility of the 6 segments as observed. No segment with the parental genotype of the Busati, as well as no segment in heterozygosity were able to produce sterility. But surprisingly, on the first time, it was observed that all segments in homozygosity produce some kind of sterility in this file. This is the 6 segments, introgressive segments of the Kefere obtained on chromosome 4 of the Buzzati. In previous studies, were more than 200 segments integrating by heterozygosis were evaluated. But from this result and reviewing the literature on introgressive segment sterility among the sister species, the Kefere and the Buzzati, we conclude that male heterozygous introgressive segment sterility occurs by a minimum threshold size of 20%. In females, this is no sterility effect. While male sterility in homozygous occurs by a minimum threshold of 2.21 to in females of 15 to males. This has allowed us to infer that each segment of female sterility in homozygosity is larger and always includes a segment of male sterility that is smaller. In conclusion, all this allows us to infer a genetic hybrid reproductive isolation architecture for this pair of sister species constituted by the heterogeneous distribution in all chromosomes of large female sterility segments that include at least one smaller male sterility segment. 2. The minimum threshold size of introgressive segments in heterozygosity to produce sterility of male is approximately 20 to 30 fertility and semi-sterility, 30 to 40 semi-sterility, and female sterility only 20% of heterozygous integration of control of complete chromosome occurred. 3. The minimum threshold size of introgressive segments in homozygosity to produce sterility of the respective sex is here for male and for both sexes. 4. The minimum threshold size of introgressive segments in homozygosity to produce sterility of respective sex is 224 or 50%. 5. The great number of female sterility genes explain the great robustness of the female fertility with strong implications for Haldane's rule. And here is the team. I'm very glad to present this work, and thank you very much.
Okay, we still have 7 minutes or so before the next talk. I don't see our speaker in the audience. So unfortunately, I don't think we're going to get answers to any questions, in which case we will just have to sit tight to wait for the next talk at 11:45 Eastern time. I was about to say the same thing that Karen just did in the chat. If there are Ian's still here, and our first speaker, Antoine. So if there's a question related to either of those talks, we've got 5 minutes to, we could happily chat about that. There's been discussion happening in the Q&A and in the chat. Ian, I see you're typing away an answer to a different question, but I was wondering if eye size some of the variation in interspecific variation in eye size may also correlate with extravagant plumage, dichromatism, or species that use visual signals in mate choice more than others. Does that also correlate with differences in relative residual eye size, I guess?
Yeah, I've not looked at sexual dimorphism. Like the Ritland data set doesn't have that kind of coverage to it. And I know people have asked about this, and as I was saying in the chat, you'd have to really be careful about your measurements because the amount of variation you're talking about is in the tens of millimeters. So it'd be a very precise measurement you're making to look at that.
Yeah, I guess like some species.
But in terms of more to your question, like in terms of ornamentation and signaling and stuff, I think that might actually get more into like the color recognition, right? And looking at how birds intraspecific variation and like wavelength recognition. So that's an entirely different dimension of the visual system. And there's a lot of, you probably know there's a lot of great visual models developed that you can implement R to look at that. And we do know there's been a lot less done on how different species respond to different color wavelengths. And there's definitely interspecific variation in that. I don't know about intra-specific, but that's a whole other kind of field to itself. But yeah.
Neat. Any other questions for either of our first two speakers while we wait for our next talk? I was wondering if instead of just selection against the metabolic costs of large eyes, that if functionally small eyes may actually perform better in a bright environment, if there's somehow, it's not just avoiding a cost, but actually there's a benefit to a small eye in a very bright environment.
Yeah, I didn't really have time to get into this, kind of the ecological side of this, the relationship to eye size and disturbance. It's been this question in the ornithological literature for a long time, especially in tropical biologists, because these species that tend to live in really dark environments are the first to disappear from disturbed areas. And there's this interesting phenomenon known as disability glare. And there's been some great experiments done where they experimentally show that individual birds become less ability to detect predators, and they show less foraging, the changes in their foraging behavior too when the light gets really bright. And there's also this phenomenon known as optical agnexia, which are basically this phenomenon where in very large eyes, the probability of experiencing glare becomes a lot more likely as well. And so the same kind of thing is like when we're driving on the highway, we experience glare in our windshield. So this was the kind of hypothesis we're presenting, is interpreting our results from Peru, saying that species with larger eyes are more susceptible to disability glare, and that's why they're less likely to exist in these lighter, much brighter environments. And I think that, you can kind of see that coming up in the comparative global analyses too, where, these species in forests tend to have larger eyes again, and potentially because, they're not experiencing bright environments. So I think getting back to your original question, I think you're spot on. Smaller eyes are probably very much adaptively, useful in bright environments in general, and more adaptable across lots of different transitional heterogeneous environments as well.
Great. I think we will let Colleen get set up for her talk now. Launching. I see another question there. Feel free to use the Q&A down there to continue the discussion. We don't want to put later speakers off schedule here. Yeah, we can see the slides. We'll just give it a few more seconds here to stay on schedule.
Okay,
We can hear you too, so we're all ready to go.
good.
So I'll introduce our next speaker, Colleen Lawless, and turn it over to you.
Thank you so much. My name is Colleen. I'm from University College Dublin in Ireland, and I'm going to talk today about one of the chapters of my PhD thesis. I'm nearly coming to the end of my thesis, so I'm getting very excited to submit and nervous. But today I'll be talking about decoding deception. And this is the molecular mechanism of cuttlefish ink on shark smell receptors. But firstly, what exactly are smell receptors? Well, simply put, these are olfactory receptors, and they're responsible for the perception of smell, and they're found in all animals. And we use them every day if we want to smell nice, or maybe if you walk into a room and you smell a nice meal cooking. However, they're very important for animals in the wild. They need them for survival. They often use them for hunting or finding potential mating partners. So how do they work? Well, olfactory receptors are controlled through membrane-bound cell surface G protein coupled receptors. So this is a large superfamily of GPCRs, and they all share a common structure. They're all 7 transmembrane. And what happens is these receptors are located in the nasal epithelium. We can see an example here in humans. And when odorants come into contact with these receptors, if the receptor recognizes the odorant, it will bind and send a signal to your brain to tell you information about your surrounding environment. So it'll tell you whether you like or dislike certain smells. And just to give some perspective of the number of these found in the genome, apex predators tend to have large numbers found in their genome. So we have the tiger and the alligator have over 1,000. Bears have around 1,800, and even birds, the turkey vulture over here has around 400. So then when you look at fish, you tend to see in the hundreds. So we have the zebrafish, the standard model fish organism, has around 200 of these encoded in their DNA. And even your pet goldfish that would spend its entire life living out of a fish tank has 100 of these olfactory receptors built into their DNA. So this brings me to my part of my PhD. I wanted to explore the OR gene repertoire in sharks. And when I started my PhD, there was very little shark sequence. There was actually under 5% of shark species had their whole genome sequenced. So studies have just kind of begun exploring this. At the minute, I think we have around 30 species, but still very low, still very poor when you look at other vertebrates. So sharks are often portrayed as having a great sense of smell. So I wanted to explore this by looking at their OR gene repertoire. And the first 2 things I have to do is I have to mine these in sharks' genomes, quantify them, and then investigate what possible odors that might bind to these smell receptors. So to mine them, to get started at the start of my PhD, I had the great white shark and the cloudy cat shark. So I downloaded them from NCBI. And in our lab, we use a pipeline called Sensomatic to mine these sensory receptor genes. And once you have a genome assembly, in order to use Sensomatic, you need a reference file. So I use annotated fish olfactory receptors that were publicly available. And thankfully, there was more other fish receptors available. There was probably over 1,000 fish olfactory receptors annotated. And the good thing about looking for olfactory receptors in the genome is that they're single exon, so they're easy to mine. And they share the 7 transmembrane structure.
Okay, I found this on the web for they shared 7 transmembrane structure. Check it out.
Apologies, my computer likes to talk to me a lot. I can't get that to go. Okay. So when I mined them, Apologies. Sorry about that. And when I mined them in the genome, I was so surprised to find that great white had only 5 olfactory receptors encoded in its genome, and the cloudy cat shark, these are beautiful sharks, they're found in coral reefs, had 4 olfactory receptors encoded in its genome. So what does this mean for sharks? Well, it means that they maintain a low number of olfactory receptors. So based on what we thought about sharks, all behavioral studies and looking at things from the media, we expected them to have a really high number of olfactory receptors, because they're often known as swimming noses. However, I wanted to identify specific chemical cues that they may favor over discriminatory capability. So my next part is, okay, so they have really small number of olfactory receptors, but what chemical cues can they actually smell? So to do this, I used in silico protein odor docking assay, and I took the amino acid sequence from the olfactory receptors from the sharks, and I put them into iTaser. It's an online server, and this will predict your 3D model of your protein. It uses TM-aligned structural alignments. And just to give an example, the good thing about these receptors is, although they're hard to crystallize, because they're membrane bound, and in sharks, it's difficult enough to do genomic studies. But because they're G proteins, their structure is similar, and they're found, this is the rhodopsin found in human, so this has already been crystallized. So this is an example of the great white shark. It's the olfactory receptor, and this was predicted using iTaser. And also what it gives you is ligand binding sites. So we need this information if we want to try investigate what chemical cues they can potentially smell. So I wanted to start with seeing if they could actually smell the chemical cue associated with blood. I think it was a very popular choice. And I used a tool called CBDoc2. But once you input in your ligand binding sites, it will predict how likely that chemical cue is to bind to your receptor. So just to give you a breakdown of some of my results, you get back a score based on your molecular docking, and this is an example here of the great white shark and the chemical cue associated with blood. And if you get back a negative score, this means that the 3D structure is stable. It's in a stable conformation. However, if you get back a positive score and the chemical cue is not binding within the ligand binding pocket, it's less likely that this will occur naturally. So when I checked the receptors, the great white and the cloudy cat shark, with the chemical cue associated with blood, I found that it binds to all of their olfactory receptors. And then I wanted to look up possible repellents, because in nature we can smell things that we like and dislike. So I wanted to investigate if sharks could possibly smell things that they dislike. And something that kept coming up in the literature for me was ink. So I thought, why would ink be used as a possible repellent? So inking is actually a chemical defense. This is the common cuttlefish. And I reached out to, I'm now collaborating with a colleague in Florida Atlantic University, Dr. Lauren. And for her PhD, she looked at inking and how it affects shark swimming behavior. And she found that when inking is put into a shark tank, it has a negative effect on their swimming behavior. So they really do not like the smell of ink. So is this an evolutionary arms race? Cuttlefish would have lost their protective shell around 160 million years ago. And with this, they would have had to come up with other ways to avoid predation. So I wanted to compare it to a known shark repellent. This is a peacock sole, and it secretes a toxin, and it's historically known as having shark repellent properties. So I download the odor for that. And we can see with the chemical cue associated with this peacock sole toxin, it had selectivity on the receptors. So it did bind to shark receptors, but not all of them. So then I compared it with the cuttlefish ink components, and I found that the cuttlefish ink binds to all of the shark receptors. So just to summarize some of my results, I found that blood binds really well to shark receptors. Sharks have a low number of olfactory receptors. However, this gives a narrow sensory space, allowing fine focused perception. However, it's that perception that cuttlefish have exploited sharks' low number of finely tuned receptors as a defense mechanism. So I just wanted to thank my two supervisors. Thanks very much for listening. Sorry for the issues there. And thank you to Lauren and the organising committee at Evolution.
Thank you. We have time for a question or two. Raise your hand or use the chat. I was wondering, do you like, would it, I'm trying to imagine what a control, so you have chemicals that like blood that you think should respond because of foraging reasons, I assume, and
Yeah.
and defensive compounds that you expect to elicit some sort of respond, or putative defensive compounds because they're defensive. Would it be useful to have some sort of a control to say, are the binding properties of either of these different from some random expectation, but it's not clear to me what that would be or how you would come up with
Yeah,
that.
absolutely. I didn't have time to include it, but there's an olfactory receptor in zebrafish, and it is found to have high binding affinity to cadaverin, and cadaverin is the odor associate. It's called the death associated odor. So I used that initially to see if the model was successful. So I got the same binding sites, and I, based on the score I got for that, I used that in my assay to know if the chemical cues had high binding or low binding. So I kind of use that as a positive control. However, I'm still trying to think of maybe negative controls, because it's kind of hard to know what they wouldn't smell if they can't smell it, if that makes sense.
Yeah, it does. Yeah.
Yeah.
Interesting. Any other questions out there? I was also wondering if interspecific among sharks might be given different life histories. Not all sharks are predatory in ways that I would think would make blood a cue, right? And
Yeah,
so,
the weird
yeah.
thing about their olfactory receptors is I've mined them from a couple of sharks, and they don't seem to be any different in terms of numbers or the type of receptors, which is really unusual, because if you look at any other vertebrate group, you'll see differences in olfactory receptors based on their life history traits or ecological habitat.
Interesting.
Yeah.
I see a question just popped up there in the chat, but maybe you could address that in the chat, and we'll get our final speaker set up to go.
Thank you.
Thanks. Hannah, you're there, I assume?
Yeah, I'm just sharing the screen.
Okay.
Is that working?
I think we're seeing your speaker view.
Nope. Wrong screen, probably. Let me try again.
Yeah.
Okay, so put it in the whole screen, so that work? Is that working now?
So that's showing your,
Oh, yep,
now
nope,
it's
there we
working.
go. Yep,
Awesome.
and right on time. So we'll introduce Hannah.
Yeah,
I don't see,
Dugdale.
yeah, I don't see your own name on. Oh, there you are at the end. Yeah, Hannah Dugdale. Take it away.
Thanks ever so much. So I'm really excited to be able to present this work today. It's work that my PhD student Ellie Chesterton did at the University of Leeds, and she's now a software engineer. And it was work with Ewan Young, who is a PhD student of mine at the University of Groningen. And Ewan is presenting in person at Montreal, and he's going to be talking about the link between reproductive traits and breast cancer using a Dutch biobank. So that's a bit of a plug for his talk. But at the moment, I'm going to be talking about single generation fitness proxies as predictors of long-term genetic contributions in the Seychelles warbler. So fitness is obviously a critical component of evolutionary biology, but it can be very difficult to measure. So fitness is, individuals with greater fitness will be those that pass on the most genes to future generations. So it's a relative measure. So if you've got individual A that you can see here, they've produced 2 chicks in the nest, and individual B has only produced 1. So in this case, individual A would have higher fitness than individual B. And in the wild, it's often very difficult to measure the absolute contribution of individuals in terms of long-term genetic contributions. So we often use these short-term proxies in terms of how many offspring they've produced in a single breeding season. But we don't know how well they correlate with individual genetic contributions. And this could be, they might not correlate very well because, for example, if we've got these 2 individuals, A and B, and we've got individual A who produces 3 offspring in their lifetime, and individual B produces 1 offspring, we would then say that individual A has got higher fitness than individual B. But it could be that individual A then doesn't produce any grand offspring, whereas individual B might produce 3 grand offspring. So the fitness metrics the fitness measures can vary depending on how many generations you're looking over. And this can vary because of many factors. So it could be due to selection, it could be migration, environmental stochasticity, and genetic drift. So one study that was one of the first to look at individual genetic contributions in a wild population was that in the Florida scrub jay. And what they did was they showed that of all the breeders that bred before 1997, 75% of those had no living descendants in 2013, so their lineage went extinct. And there was a lot of skew in terms of those individuals that did contribute to the 2013 cohort, in that 6 breeders were ancestors of 25% of that cohort. And using gene dropping, they're able to show that this was likely due to genetic drift. So reproductive output might not predict long-term genetic contributions because of that example due to genetic drift, but also due to factors such as a quantity-quality trade-off. So individuals might produce many offspring of poor quality. There could be density, frequency, and environmentally dependent variables, and migration may play a role. But also, when we're going out in wild populations, it might depend upon the stage at which you are measuring fitness in the offspring when you're counting them. So this bird here, you can see, has got 2 chicks in a nest. But actually, when we went back a few days later, there was only, unfortunately, 1 dead chick in that nest. And therefore, whether you count offspring as nestlings or fledglings or those that recruit to the first year can differ, can give you different fitness measures. It's also difficult to measure fitness in the wild because populations are often open and you have immigration and emigration of individuals. So those birds that immigrate in, you don't know about their reproductive success before they join the study population. And individuals that emigrate, you don't know if they've died or if they're breeding elsewhere. And you also need to measure the lifetime reproductive success of individuals, so over multiple breeding seasons, which can be difficult to do in the wild. And you need to account for promiscuity, often using genetic markers to determine the genetic pedigree. So this is where our long-term Seychelles warbler data set comes in, because we've got long-term data spanning back to 1985 when the ringing originally started, And the intensive effort started in 1997. And this is the island of Kuzan, where the main study island of ours is. And it's a closed population. So you can see in this photo, Kuzin Island is the closest in the background, but birds virtually never move between these islands. And because we colouring birds, we have very high resighting rates each year. So we know if we don't see an individual that it has died. So we've got very accurate survival data. We also have very accurate reproductive success data. So this is an aerial view of Kuzan Island. And we monitor all the roughly 115 territories in the 2 breeding seasons each year. And so we follow all the breeding activities, and we collect blood samples from individuals to assign genetic parentage. And this is genetic pedigree that we have. We've got over 2,000 individuals. And you can see, hopefully it might take a little bit, it might be a little bit slow loading, but you can see the dots are the individuals. Red line goes from a mother to offspring, and overlaying those are the blue lines from the father to offspring. Now, in the early years, blood wasn't collected all the time. Not really, it didn't really start until 1995. So we've got low parentage assignment of about 60% before 1997, but after that it's 94%. So what we did was we took these, I think, 11 cohorts, which totaled 900 individuals, and we calculated their individual genetic contributions over 3 generations, which is 15 years in the Seychelles warbler. So for a bird born in 2007, we looked at their individual contributions in the 2022 cohort. So when we've got these IGCs, what we then did was to ask how well these 6 fitness proxies correlate with the IGCs. So we looked at whether an individual gained a dominant breeding position or not, because birds mainly breed as dominants, but females may also co-breed. We looked at the age at which they first bred, how long they were a dominant breeder for, what their lifespan was, and then we took 2 measures of lifetime reproductive success by either counting the number of offspring that survived to independence, which is 3 months in the Seychelles warbler, because it's a lot of post-fledgling feeding, or the number that survived to adulthood at 1 year. And these are results. There's going to be 6 of these graphs, and on all of them, the single generation fitness proxies on the x-axis. So here, this is whether or not they obtained a dominant breeding position, and the individual genetic contribution is on the y-axis. And what you can see is that there was no correlation between whether an individual gained a dominant breeding position or not and individual genetic contribution. But it was a very strong predictor of lineage extinction. So 98% of those birds that never gained a dominant position, their lineages went extinct after 3 generations. And this is because dominant males are the only ones that gain paternity pretty much, and only about 11% of offspring aside, have a subordinate co-breeding female as their mother. And a lot of those co-breeders go on to gain a dominant breeding position. The next metric we looked at was age of first breeding attempt, and there was no correlation here, and there's quite a large distribution in that age of first breeding. And then we went on to look at how long an individual was a dominant for, and there was a strong positive correlation here. So individuals that had a longer dominant breeding tenure had higher individual genetic contributions. And they also, it was also associated with lineage extinction. So those individuals whose lineages went extinct had a mean tenure of 2 years versus 6 years for those individuals whose lineages did not go extinct. We find a similar relationship with lifespan. So there's a positive relationship, and again, it predicts lineage extinction. So those individuals whose lineages go extinct had a mean lifespan of 3 years versus 8 years for those whose lineages did not go extinct. And for lifetime reproductive success, we found a positive relationship again. And again, it's a good predictor of lineage extinction. So individuals whose lineages went extinct had a mean lifetime reproductive success of 0.4 versus 4.6 for those whose lineages did not go extinct. This was looking at individuals' offspring that survived to at least 3 months. And if we look at individuals who survived to at least 1 year, we've got a very similar relationship and also prediction with the probability of lineage extinction. So of those metrics that short-term metrics that predict individual genetic contributions, what is the predictive power of them? We found that dominance, breeding tenure, lifespan, and lifetime reproductive success. I'm just showing that for adult offspring here, but it was also for those that survived to 3 months. They all predicted individual genetic contributions, and we ran the model separately for males and females. And what you can see here is that for the individuals that gained a dominance, breeding tenure, looking at how long that tenure was, there was a 31% correlation with individual genetic contributions for females and slightly higher for males, but there was a lot of overlap there. And this is potentially linked to females co-breeding as subordinates. There was a greater predictive power slightly for lifespan, and it was similar between the sexes. And for lifetime reproductive success, the predictive power went up, and it was surprisingly high, 0.52 for females and 0.67 for males. And that's potentially due to males having much more variance in reproductive success. So how does this compare to other studies? I've mentioned the Florida script jay, but Jane Reed has also worked on this in song sparrows. Ewan's looked at this in a pre-industrial Swiss human population, and it's also been looked at in house sparrows and bighorn sheep. And they've looked at different numbers of generations, and not looking at short-term fitness proxies, but looking at lifespan and lifetime reproductive success. And you can see overall that as the data on survival and reproductive success increases, then the predictive power of these fitness proxies increases. So in summary, what we can see is that the short-term fitness proxies that we looked at as whether an individual gains a dominant breeding position or their age of first breeding didn't predict individual genetic contributions, but they can predict lineage extinction. So we had very high predictive power looking at whether an individual gained a dominant status in its life or not, and lineage extinction. And overall, I didn't mention this earlier, but there was a high probability of lineage extinction in the data set. So 72% of individuals born before 2007 didn't contribute to the 2021 gene pool. And that corresponds to a similar number of generations in script jays, where it was 75%. And in Ewan's study in humans, he looked over 10 generations and it was 73%. And overall, what we found is the predictive power of the fitness proxies increased as their information on survival and reproduction increased. So thanks for listening, and I'll be happy to answer any questions.
Thank you. Happy to take any questions from the audience. Use the Q&A or the chat or raise your hand. I'm curious, is that a consistent result across the other studies that the correlations with long-term success were stronger in males than in females? Or is that unique to your system?
That's a good question. I'd have to actually go back to the papers to look at if they have separated that. I think Ewan definitely did separate that in the human data set, but off the top of my head, I can't remember. Yeah, I think it will depend upon the degree of reproductive skew in those species as well, I think. Yeah.
Well, if you had enough studies, you could correlate it with the degree of reproductive skew potentially to ask if that is the causal agent underlying the difference. Yeah. It certainly makes one worry about the wealth of studies out there using proxies for fitness that are potentially.
Yeah, well, it's difficult to know because some of these, the individual genetic contributions could be affected very much by genetic drift, which is what we suspected in this Seychelles Wolver population, because it's a closed island and the total population size is about 320. So we expected we actually thought there'd be lower correlation with individual genetic contributions. So we were quite surprised that we could explain so much variation. And it comes down a little bit to whether or not the IGCs are stabilized, and they have stabilized in some cohorts and not in others. So it might the predictive power might decrease depending on how many generations you look over as well. But for us, this is a very long-term data set. So although we've got data going back to like 1997, yeah, we could only look over 3 generations.
Yeah, yeah. It's an impressive data set. My understanding is there's no final talk in this session.
Oh, yes.
So it's all right that we've run a tiny bit over. I don't see any other questions popping up. If there aren't, then it'll be a short break before another set of sessions starts in a little bit. And
Thanks
thank
for
everyone
sharing.
for coming. Yeah, it was an interesting set of talks.
Thanks, everyone. And thanks, Howard, for moderating.
Happy.